All right, so our next lecture is on blood. We talked about hormones, which are chemical signals that travel in the blood. Sorry for the lateness of this lecture, but we've been without power for a little while here. Um, like I assume many of you in Lubbock have experienced. Whatever happened yesterday, whatever the power things and LPNL, turned off our power for a little while so they could fix something. I don't know, it's friggin' hot. So, right now, we're going to talk about blood. The first things, what does blood do? It is our distribution medium. Oxygen, carbon dioxide, waste products, and as we said, hormones. It's also involved in regulating body temperature, circulation to the skin, for instance. It is temperature based, and body temperature goes up, blood flow to the skin goes up, so that heat radiates away from the body. Also, one of the functions of blood is to protect against leaking out of your body. So it's involved in stopping itself from leaving, and it circulates the immune system, which is for another day. So blood is a connective tissue. It's the only fluid tissue, but it is connective tissue. And like other connective tissue, is made up of cells and an extracellular matrix. The extracellular matrix for blood is plasma. So this is that ground substance and proteins outside the cells. The formed elements are the cellular component of blood. Now, of these formed elements, only white blood cells are intact cells. Red blood cells don't have a nucleus anymore, they're not intact cells. Platelets are just chunks of a bigger cell. Like I said, blood is the only fluid tissue in the body, but it is connective tissue. So here again, the formed elements of blood. Now this is not a representative slide, meaning that if I took your blood and threw it on a microscope slide, it would not look. But this is a drawing that illustrates all the different types of formed elements. Red blood cells, the different types of white cells, and platelets. Now if we take your blood, we put it in a little vial, we spin it in the centrifuge really fast, what happens is the formed elements get shoved to the bottom because they're heavy, and the plasma will float to the top. From this, we can take a reading called hematocrit. And hematocrit is the percent of blood volume that's made up of red blood cells. It's around 47%, or take 5% for males, and around 42%, give or take 5% for females. Here we see an illustration of this idea of hematocrit. So now that it's bottom, we see the red blood cells. Right there in the middle, that little white layer is made up of white cells and platelets, and then plasma floating to the top. You've seen blood. It's red. pH of blood ranges between 7.35 and 7.45, depending on where we are in circulation. And its average temperature Celsius. Makes about 8% of your body weight, and you've got about 5 liters of it. The plasma is the extracellular part of blood. And here we'll have proteins and enzymes and stuff that's dissolved in the plasma. The fluid part of that, like the actual fluid minus all the proteins, serum. Now in plasma we've got proteins and we have the albumins. The albumins are the most abundant of the proteins. They contribute to the thickness of plasma and the osmolarity of plasma, which is this pull on water. So in that way it's going to influence blood pressure because the more fluid you have in the blood, the more pressure you have in the blood. Also, it's going to affect balance of fluid in terms of movement of fluid. For instance, the kidneys. The globulins are antibodies and part of your immune system. More on that later. Finally, we have fibrinogen, which is the precursor of something called fibrin that makes blood clots. Now, this is the first time we've seen this idea, fibrinogen. When you see that suffix there, that gen suffix, what that is going to indicate is an inactive protein. Anytime you see that, whether it's fibrinogen or pepsinogen or trypsinogen or any of this, all of that is an inactive protein. It means that it has to be activated to do anything. So fibrinogen is inactive. It's soluble. It's in your plasma, not doing anything all the time. When it's activated, it becomes fibrin. We'll talk about that process of activation a bit later. And fibrin is going to help form this blood clot. These plasma proteins will be formed by the liver, except for the parts that are parts of your immune system, the globulins. We'll talk about that whole plasma cell and B cell thing later. Also in plasma, we've got things like nitrogenous waste from amino acid breakdown, the end products of protein catabolism, and these will be removed by the liver and the kidneys. There will be some nutrients dissolved in the plasma, and a little of the blood gases as well. Lots of electrolytes, most of it's sodium. And this is, again, going to contribute to blood's osmolarity, this pull that blood has on water. So like I said earlier, only the white blood cells are complete cells. Red blood cells, nothing inside but hemoglobin. They're just sacks of hemoglobin. And the platelets are chunks of a bigger cell. With the exception of red blood cells, most of the formed elements don't last very long in the blood. And most of these blood cells will come from the bone marrow, specifically red bone marrow. So, just as a review, you find red marrow in the adult, uh, in the diploid of flat bones, and in the proximal epiphysis of the femur and the humerus. Talk about red blood cells. Red blood cells are biconcave disc, which means it looks like that. There's that there. There's virtually nothing inside except for hemoglobin, which is gas transfer. This is a great example of this idea of form and function complementing one another. This is sort of a big overarching theme for anatomy and physiology, is this unity of form and function. And the red blood cell is a great example of that. The shape of the red blood cell, this shape, provides the maximum amount of surface area per volume of the cell. And that surface area is important because it allows for that movement of the blood gases in and out of the red blood cell. It also allows for the red blood cell to change shape and sort of scrunch up and fit in different places in tiny little capillaries, even in and out of the bloodstream in special places like the spleen. Most of the cells in your blood are red blood cells. If we go back and we look at that hematocrit, there's your red blood cells. 45% of blood, period, is made up of red blood cells. But nearly all the cells of the blood are red blood cells. Less than 1% of your blood is white cells and platelets. Here are red blood cells on a needle. So again, surface area to volume ratio based on that biconcave shape. Screw that up, and you disrupt blood's ability, or the red blood cell's ability to do its job, because its function is so reliant on its structure. And we'll look at some structural abnormalities of the red blood cell. We'll look at some pathophys a bit later. Like I said, discounting water, nearly all the red blood cell is hemoglobin. It's just a giant sack of hemoglobin. They don't have mitochondria. You want them to carry oxygen, not to burn it. So any ATP production is just glycolysis. The job of the red blood cell, gas transport. Hemoglobin binds to oxygen. Oxyhemoglobin. Hemoglobin looks like that. 1, 2, 3, 4 chains. 2 alpha, 2 beta chains with an iron atom suspended in the middle by this heme group, which means that every hemoglobin protein can carry 4 oxygens. That heme group. Hemoglobin picks up oxygen in the lungs, oxyhemoglobin, and we drop it off in the tissues when we get deoxyhemoglobin. Oxyhemoglobin is bright red, and deoxyhemoglobin is slightly less bright red. Carbon dioxide can also bind to hemoglobin as carbaminohemoglobin. About 20% of carbon dioxide in the blood travels this way. Where do red blood cells come from? Well, in the embryo, we have the yolk sac, the liver, the spleen, even the thymus. The liver stops producing blood cells when you're born. The spleen and the thymus will keep up with white blood cells. We've got these lymphoid things going on. But normal physiology here, discounting any development. Red bone marrow is where red blood cells are going to come from. And most white blood cells and platelets as well. We have stem cells, hemocytoblasts, that are continually multiplying, and they will differentiate into different cell lines. There are different hormones, chemical signals, that trigger this process. So here's the big picture hemopoiesis. You can see our stem cell here differentiating. Now, the first thing we're going to talk about is this one up here. The formation of red blood cells. That is erythropoiesis. Erythropoiesis produces about 2.5 million red blood cells every second. The first of those is called a proerythroblast, and that cell has receptors for a hormone called erythropoietin. Erythropoietin, or EPO, is produced by the kidneys. When the kidneys decide that they're hypoxic or there's not enough blood flow, they respond by releasing EPO. EPO tells your bone marrow, these proerythroblasts, to multiply so that you get more red blood cells. You've probably heard of EPO because of Lance Armstrong. So Lance Armstrong, the Tour de France wins. Cheating. But hey, you know, you do what you got to do. So how he's cheating? He's cheating by using erythropoietin, or EPO, which causes you to make more red blood cells. If you make more red blood cells, your blood carries more oxygen. If your blood carries more oxygen, your muscles can make more ATP, because that's how you have to make ATP. You take oxygen and glucose, and you get ATP. So now you've got more oxygen. Downside of that, red blood cells are the major contributor to viscosity. So the more red blood cells you have, the thicker your blood gets. And as your blood gets thicker, it's harder to move through your bloodstream. We increase the viscosity of the blood. This condition is called polycythemia. That is an elevated number of red blood cells. This is dangerous because at some point your heart might just stop. So why would a professional athlete use erythropoietin? Obviously to win. Winning, dying, you do what you got to do. Risk benefit. Now, in our production here, we go pro-erythroblast, erythroblast, and we start throwing out organelles. By the time we get to this stage, this reticulocyte, the organelles are pretty much gone. There's a degrading endoplasmic reticulum. And some of those reticulocytes will actually be found in the blood. You'll see more reticulocytes in the blood if you've lost blood recently. We kind of rushed through. From start to finish, this process takes about 15 days. Like I said, blood loss will speed up that process and increase the number of reticulocytes circulating in the bloodstream. Hypoxia, a lack of oxygen carrying capacity in the blood, happens because you don't have enough red blood cells. So we have this balance between red blood cell production and production. Your average red blood cell lives about 100 to 120 days. So they're constantly dying and they're constantly being produced. Again, your direct stimulus, erythropoietin, the major hormone that tells you to make blood, red blood cells. So why would you have hypoxia? You're bleeding, your blood cells are dying, your blood cells don't have enough hemoglobin, or there's not enough oxygen. All of those things can cause hypoxia. Now, let's say you live at a higher altitude, like Denver. There's not as much oxygen there. If you've ever been to Denver, it's kind of cool, and it kind of sucks. It's hard to breathe, but people live there because you adjust to it. If there's not enough oxygen, your body will make more red blood cells to compensate for it, so that your blood will carry more oxygen, even though there's less of it in the atmosphere. So erythropoietin, more red blood cells. Other things that enhance erythropoietin, testosterone. We go back and we look at hematocrit, right? The hematocrit for males is a little higher than it is for females because of testosterone. And this is another way that Lance Armstrong was cheating, injecting himself with testosterone, which also increased the amount of erythropoietin in his blood. So here's our fun seesaw thing with the homeostasis of red blood cell production. That is negative feedback. As your red blood cell count goes down, can either lead to erythropoietin. With that release of erythropoietin, you make more red blood cells. Stops the release. All right. So what do you need to make red blood cells? Obviously iron for hemoglobin. Iron is not super easy to absorb because it's metal, and you're constantly losing it. Stomach acid has to convert to iron to an absorbable form, and that absorbable form has to be transported to the small intestine and the liver, and there are other things as well. The vitamins, B vitamins, B12, B6, I think is folic acid. I can't remember what number folic acid, but B12 and folic acid are involved in cell replication of red blood cells in that erythropoiesis. There are enzymes involved, and vitamin C and copper are cofactors in some of those enzymes. So there's lots of nutritional requirements here. Now, as we talk about this idea of B12 and I suppose folic acid, we have this misconception of how this process works. B12 is essential for rapid cell production. If you have a lack of vitamin B12, erythropoiesis will be slowed down to the point where you're anemic. But adding more B12 does not equate to making more red blood cells, assuming that you have normal physiology. So if you have currently an adequate amount of red blood cells, and I add B12 to it, that doesn't mean that you're going to make more red blood cells. If that were the case, B12 would not be sold over the counter, and it would be a performance-enhancing drug. It is not. Bringing more B12 into your body does not equate to your blood carrying more oxygen or making more red blood cells, because that would be dangerous. Just like putting more gas in your car does not mean that your car has a higher top speed now. There is this misconception that B12 equals energy, and that's not the case. We'll talk more about B12 when we get to vitamins, but most people are not B12 deficient. Now, there are exceptions, uh, but the vast majority of us have enough B12, and adding more B12 does not equate to making more energy, carrying more oxygen, or anything else. It just means that you're going to lose more B12 in your urine because you brought too much in. So more on that later. Here's iron absorption, and I'm not worried about the molecular biology of ferritin and apophyrin. 120 days. They don't have a nucleus. So 120 days in, they bouncing around your bloodstream. They can't fix themselves. So they're going to die. This is where your spleen comes in. Macrophages in the spleen will latch onto these and break them down. They get recycled. The heme, the globin get pulled apart. We recycle as much iron as we can, although some of it's going to be lost to the feces. One of the byproducts of the breakdown of hemoglobin is bilirubin, which is a yellow pigment, which gets put into bile and then secreted from the body, excreted from the body. When your liver fails, and you can see this here, the liver, when your liver fails, none of this is going to happen. And so that bilirubin will build up in the body and you'll turn yellow. It's jaundice. All right, so moving from red blood cells to white blood cells, less than 1% of total blood volume is white blood cells. And part of this is because they leave. They'll leave the capillaries through a process called diapedesis, where they kind of squeeze out and then like blob it. So they move through the tissue spaces to this amoeboid action. Leukocytosis, too many white blood cells account over 11,000 per cubic millimeter. We would expect to see this in any sort of infection. So here we see our formed element potential count. Now we have two major groups of white blood cells, the granulocytes and the agranulocytes. The granulocytes are named for dark staining granules in the cell. Agranulocytes don't have that. Here's a nice little mnemonic to remember the white blood cells from most to least common. Never let monkeys eat bananas. It's not advice. You should give the monkey the banana. But it is a nice little mnemonic to remember this ranking. Neutrophils, lymphocytes, monocytes, eosinophils, basophils. Neutrophils are the most common. Basophils are the least common. The granulocytes are phagocytes, which means they eat other cells. They include the eosinophils, the basophils, and the neutrophils. The agranulocytes include the lymphocytes and the monocytes. So the neutrophils, the most common white blood cell. So what's the most common use of the white blood cell? To kill bacteria. Inside we've got these granules that have enzymes and chemicals to attack bacteria. Eosinophils are going to be involved in the parasitic reaction, and they're going to help modulate the rest of the immune response. They can cause immune action that can actually be harmful to type of asthma called eosinophilic asthma, where the chemicals released by eosinophils cause the bronchi. Basophils contain histamine. Basophils are the rarest of the white blood cells. And part of that is because there are other cells called mast cells that do the same thing. So there's a neutrophil, an eosinophil, and a basophil. The lymphocytes don't have those granules, but there's not a lot of lymphocytes circulating in the blood. Most of them are in the lymphoid tissue, called lymphocytes. The lymphocytes are what we think of as your immune system. Immunity meaning that you have this memory that occurs. And there are two types of lymphocytes, T cells and B cells. T cells are going to act against cells that are infected with viruses or cancer cells, abnormal cells. B cells are going to create a cellular line that ends up producing antibodies. More on this when we get to the immune system. Monocytes are the biggest of the white blood cells. Monocytes will leave the bloodstream and turn into macrophages, where they become phagocytic. Again, more on this one, immunity. So there's a lymphocyte and a giant fat monocyte. White blood cell production is called leukopoiesis. The hormone that stimulates leukopoiesis is called a colony-stimulating factor. And those stem cells possess receptors for these colony-stimulating factors. Colony-stimulating factors are produced by white blood cells in response to infection. Now we have positive feedback. So a white blood cell that encounters infection releases this colony-stimulating factor, which comes back and causes the production of more white blood cells. Some of the white cells will leave the bone marrow before they're done, and they'll go to the thymus to grow up. These are called T cells. Again, more on this later when we get to immunity. White blood cells aren't like red blood cells. You don't want them in the bloodstream very long. The granulocytes, about 5 days. The monocytes will leave in about 20 hours. The lymphocytes may be in your lymphatics for decades, but not in the blood circulating. So here is this whole production of white cells. And again, I'm not worried about the minutia here in terms of myeloid versus lymphoid stem cells. Platelets involved in blood clotting, like I said earlier, are just chunks of a bigger cell. It's called a megakaryocyte. And little pieces of it break off to form platelets. Platelets, again, secrete clotting factors that are involved in stopping blood loss. Platelet production is called thrombopoiesis, and the hormone here is thrombopoietin. Thrombopoietin from the liver to the kidney triggers the formation of these megakaryocytes, which replicates the DNA without dividing and then just breaks off into tiny little platelets. Live for about 10 days, although a bunch of them will be stored in is thrombopoiesis. The job of the platelet is to stop bleeding, hemostasis. This is very effective in stopping blood loss from small blood vessels. All the mechanisms here will involve platelets. So when you damage a blood vessel, it's going to very quickly try to stop that. The first thing that's going to happen when you damage a blood vessel is that blood vessel is going to constrict, a spasm. Pain, is involved here, and it causes constriction of the vessel, which is going to minimize blood loss. Now, this is not a fix. It's not going to completely stop it. It's going to minimize it. Basically, what this is doing is buying time for an actual stoppage of the bleeding to occur. Platelets are going to plug the hole. Now, normally the inside of your blood vessels is super smooth. It's coated with prostacyclin. Prostacyclin is one of those icosanoids, and it's like blood vessel Teflon, so that nothing sticks to the inside of your blood vessels. But when the blood vessel is broken, it exposes collagen fibers. If you take a piece of paper and you tear it, and you look at the edge of that rip, you'll see little bitty fibers sticking out of the paper. Much the same as you would in a blood vessel. You tear that blood vessel, you'll see little bitty collagen fibers sticking out, and it's rough. And those platelets stick to the collagen fibers. Once those platelets stick to the collagen fibers, they degranulate. They release serotonin, which causes the blood vessel to constrict. They release ADP, which attracts more platelets. Now we're in positive feedback. More platelets are attracted, more ADP is released, more platelets are attracted. We also release thromboxane, another icosanoid, which produces or promotes those platelets to clump together in more vasoconstriction. Again, this is a positive feedback cycle. So there's our vascular spasm here. Platelets, that. Recruiting more platelets. And finally, our last step is the formation of the proper blood clot, coagulation. This is a set of reactions that turns blood from a liquid into a. This is magic to watch. When you see it, what you see is this liquid, it's fine, and then all of a sudden out of this liquid, this mesh forms, and it's out of nowhere. Because we have that inactive protein, fibrinogen. Fibrinogen is in the plasma all the time. It's just floating around. It's not doing anything. It's inactive. And when this happens, chemical reaction, we get an enzyme called thrombin that turns fibrinogen into fibrin. Now, this is a complicated reaction. It looks like this in phase 1. Yeah, there's multiple phases to this crap. Do I care that you know this? Absolutely not. You don't need to know that the intrinsic pathway involves this factor 12 and 12A and 11 and 11A and calcium being involved. This is not what I care about whatsoever, nor should it be what you care about. This intrinsic pathway is triggered by platelets. The extrinsic pathway is triggered by trauma to the cells that make up the blood vessel. Regardless, these are going to progress in this chain reaction toward this prothrombin activator. That prothrombin activator then catalyzes prothrombin to become thrombin, which then turns fibrinogen into fibrin. 3 phases, man. To get to this fiber, this insoluble polymer. And there's this factor 13 that's involved to cross-link that fiber. Why? Why is this so complicated? Look at this. The answer to that is, do you really want this to happen if it doesn't absolutely have to? No. No, you don't. The last thing that you need to happen is this just to go off randomly inside the bloodstream. Because then you get clots forming where you shouldn't get clots forming. So there's a lot of complexity here, and all the pieces have to be in place for blood to clot correctly. If any of these things are missing, these little clotting factors, yeah, blood's not going to clot right. So we're not going to get into this whole factor 10 again, this different phases here getting toward this. What I'm concerned with you knowing is this down here at the bottom, this fibrinogen turning into fibrin, which is triggered by that thrombin enzyme. That's the important enzyme. That reaction right there. Here's what it looks like. Notice that there are red blood cells that are caught in this mesh because right before this, everything's cool. All of this would have been liquid plasma. The red blood cells are floating along. Everything's cool. And then, bam, that chemical reaction happens, and this mesh just forms out of nowhere, trapping those cells in there. Now we get a blood clot, which stops the bleeding. As soon as that happens, the clot's going to start to retract and pull the broken edges of that blood vessel together. Chemicals from the platelets are going to stimulate that blood vessel to repair itself. We're also going to have another enzyme, kallikrein, which converts plasminogen into plasmin. There's your suffix again. That's an inactive protein floating around in the plasma. It's converted into plasmin, which breaks apart the blood clot so that we start to dissolve it. As the cells replicate and the blood vessel repairs itself, that blood clot's going to be dissolved. This is also positive feedback. We've got two big mechanisms here that keep blood clots from getting too big. One, those clotting factors are immediately going to be removed and spread out. They all have to come together in the same place at the same time to work, but then they get distributed around, and now it's not going to happen. This enzyme is caught in that fibrin thread, and it can't go anywhere else. So the reaction is limited to the area of damage rather than being spread throughout the body. You don't want all your blood to clot. It's also problematic. So you get cut, this reaction happens, stops the bleeding. Get cut again, this reaction happens, stops the bleeding. Get cut again, this reaction happens, stops the bleeding. There's only so many times this can happen before you can't stop the bleeding anymore because it's all busy. All these factors are currently busy stopping bleeding in any of the other number of injuries you have. And now this last one doesn't stop bleeding. It's all busy. Sorry about that. So, heparin, these anticoagulants will inhibit thrombin activity so you don't have abnormal clotting. Normally, you don't get clotting because platelets don't stick to the walls of the blood vessels because of prostacyclin, blood vessel Teflon. On occasion, you may find yourself in need of a blood transfusion. Whatever life choices you've made have led you to this point where you suddenly need blood. We can give you whole blood. That is, somebody else's blood or even your blood that you've saved somewhere. We could give you packed red cells. We could give you platelets. We could give you just plasma. Transfusion of somebody's blood is tricky because you need it to match. Now we get into this area of blood typing. So, how does this work? Well, on your red blood cells, there are at least 30 types of antigen. Now, an antigen is something that cells or chemicals, products of your immune system can bind to. And there are two major types of antigens. There are self-antigens and non-self antigens. You can think of antigens as like a gang tattoo on your cells. There's an obvious Crips and blood joke here because we're talking about blood, but I'm going to pass on that because I'm better. So, gang tattoos on your cells. So you got your red blood cell, and it's got its, whatever, West Side tattoo on it. It marks your cells as yours. So now when your immune system's going by and checking stuff out, it knows that's your cell. Now, somebody else's red blood cell has whatever, pick another gang tattoo on it. There. Now your immune system goes by, it sees that, and does like an immune system drive-by and kills that cell. It's not that simple, but just roll with it. This is an antigen. Those markers on the cell membrane, anything, it doesn't even have to be in a cell, that your immune system bind to or recognize. Now, your red blood cells, there are at least 30 types of these little markers on the cell, and they're really kind of unique for you. What happens is we have antibodies in your bloodstream that will stick to that little marker if it's not right, and it can stick to, a bunch of those little markers. What will clump those cells together? Something called agglutination. Agglutination is this clumping together of cells where we clump them all together and we keep them out of circulation. Then that gets destroyed by a macrophage. We classify your blood type based on the presence of those antigens. What are blood types? The ones that we care most about are the ABO and the Rh group. They cause the most vigorous reactions to hardcore coagulations, agglutination of cells. There are other blood types. There's a few that are weak agglutinogens. We care about them, kinda. I mean, ideally, we'd match them all. Ideally, if I was going to transfuse blood into you, it would be your blood. Life isn't always ideal. So sometimes it has to be somebody else's blood, and at the very least, I've got to match these. If I don't, I might kill you. If I don't match these, I'm probably not going to kill you. It's not going to be uber comfortable, but you do what you have to do sometimes. So you don't always get to match them all. The ABO groups. So the ABO groups give us 4 major blood types: type A, B, AB, and O. And this is based on the presence of 2 agglutinogens, the A and the B antigen on the surface of the red blood cell. Your blood will have antibodies against other blood types. So if you have type A blood, right, your blood cells have the type A gang tattoo. You freaking hate type B. You have anti-B antibodies. I put B blood in there, you get that gang warfare. So now how does this come about? Well, it's genetic. Types A and B are what we call co-dominant. Co-dominant alleles, which means the A is a dominant, but the B is also a dominant allele, which is how we get type AB. So if you have a person who is type A and the person who is type B, you're a little square, At the very least, you can get one right there that's type AB. But the O here is recessive, which means that if you have type A blood, your genotype may be AO. Type B blood, it may be BO. So this would still give you type B blood, and this would still give you type A blood. We do our little square here, we get AO, BO, OO. In order to have type O blood, this has to be your genotype. You have to get an O gene from either parent. To have a type A blood, you just have to get one type A gene. Type B, you have to get one type B allele. AB, you've got to get one of each. Type O, even though that's the recessive allele, is easily the most common blood type in the United States. Type AB is the least common blood type in the United States. Here's this. So let's start at the bottom then. So if you have type O blood isn't a type. Type O blood means that you don't have any of those gang tattoos on your cells. You're a complete civilian. What are gangs? You've got nothing. Tattoos? Never. So your cells are totally clean, walking around completely oblivious. And you have antibodies against both A and B. You're not putting up with any of that gang crap. Now, 45% Asians in the United States, type O blood. About half of African Americans, 40% of Asian Americans, and like 80% of Native Americans, type O blood. Type A blood. So now you got your little A gang tattoo there. Hate type B, anti-B antibodies. 40% of Caucasians, about 27% of African Americans, about the same for Asian Americans, and about 16% of Native Americans. Type B blood, a little less common than that. You can see, same principle though. You have your B antigen on the cells and your anti-A antibodies. And then you have type AB blood. Type AB blood is like when you go get out both the Crips and the blood tattoo, just cover all your bases. You're cool with everybody. And people with type AB blood, there's no antibodies. What that means for you is that if you have type AB blood, you are the universal recipient because you don't have any antibodies. You like everybody. You're cool with all of it. If you have type B blood, you can get type B or type O. Type A, you get type A or type O. Type O, you can only get type O. Now, anybody else can get your blood, but you can only get type O. It kind of sucks until you realize that, especially in the United States, friggin' everybody else has type O blood too. So it's not like it's a rare blood type. The RH group is what gives us that positive or negative on the blood. There are like 45 different RH agglutinogens. The only one that we care about really is this factor D. So if you have a positive blood type, which most likely you do, I want to say it's like 80% of Americans have a positive blood type. That's factor D. Now the RH factor for factor D is a simple dominant recessive. RH positive is dominant, and RH negative is recessive. So you have to inherit two recessive alleles to get that negative blood type. So it's a lot less. Now, the thing with the RH factor is that you don't necessarily have antibodies against it. So if you have a negative blood type, you don't immediately have these antibodies. Now, if you have type B blood in this case, you immediately, yeah, we've got those anti-A antibodies. You inherently hate type A. But if you have a negative blood type, you don't inherently hate positive blood type. You have to be exposed to it first. And if you're exposed to it, now you hate it. And the second exposure to that positive blood type, Now you get agglutination, you get a transfusion reaction. This is most problematic in pregnancy. Because of the way the genetics work, it's very perceivable. It's common even for a woman who has a negative blood type to be carrying a baby that will have a positive blood type. So, right, if you have a negative blood type, again, that negative blood type is recessive. So you have to have two negative alleles. If you have a positive blood type, you just have one positive allele. So there's a positive there. It doesn't matter what the other one is, even if it's negative. The chances of you having a uh, positive baby, even in the case scenario, so that uh, is 50%. Worst case scenario, it's 100% if your spouse have a positive blood type. So now what happens is that you have a negative blood type, the baby's positive. Antibodies cross the placenta. And so those Rh antibodies, the anti-Rh antibodies, will then attack the fetal blood. And they will destroy the red blood cells in the fetus. This is called hemolytic disease of the newborn. So if you have a negative blood type, when you're pregnant, they treat you with Rogam, which keeps you from being sensitized. It prevents that from happening so that your antibodies don't attack the blood cells of. If you give somebody the wrong blood type, now when you mismatch these ABO groups, there's a high likelihood that you're going to kill them. Why? Because the donor cells get agglutinated. They're going to clump together. They're going to clog tiny blood vessels. The first thing that you'll see is called petechiae. See, I'm not even going to try to spell it. Like little bitty flowers right under the skin as the capillaries begin to rupture. They're going to itch like crazy at this point as well. Those little blood vessels rupture. The blood cells that are agglutinated together rupture, and hemoglobin leaks out into the blood. So now you haven't helped anything, right? Your idea of giving someone a blood transfusion, they can carry more oxygen, they're not hypoxic. Well, that sucks because you didn't help anything. So you're still going to be hypoxic. And now you got free hemoglobin in the blood, and that's going to clog up the kidneys, and then you have acute renal failure. Apologize for the barking in the background. That is a corgi. He's loud. Full disclosure, adorable dog. And we'll talk about the adorable dog when we get to the liver, because there's a whole thing. Anyway, you'll get to see a corgi. Look forward to seeing a puppy. All right, so blood typing. We're going to do this in just a minute. Say we, I mean me, because we're not actually in lab. But anyway, what I'm going to do, I'm going to take blood, obviously, probably my blood, depending on which kids are asleep, probably my blood. Um, and we're going to add it to the antibodies to do blood typing. And a positive reaction gives you the blood type. So like this. So if you had type A bleeding, right, and clot in both of them, type A, just and then here's type O, you get nothing. More on this in just a minute when I do the blood typing thing. Bleed a lot, it's restore volume. Now, here's the thing. This is called hypovolemic shock. Low volume. Hypovolemic. So here's the thing. Fluids move with a pressure gradient, including blood. Pressure is based partly on volume. And if you don't have enough volume, you don't have enough pressure. And if you don't have any pressure, you have no circulation. And if you don't have any circulation, blood's not moving, and you're screwed. If your blood doesn't move, why is your heart even wasting its time beating? It's not moving any blood anywhere. So it's very easy to die. So it's important that you replace the volume. Even if you don't replace the oxygen carrying capacity of the blood, the volume has to be replaced. It can be replaced with saline. It can be replaced with plasma. Side note, plasma. So when you go to some place to sell your plasma and you get like 30 bucks, like you're saving lives. Not in the way that you think you are. First of all, don't say that you're going to donate plasma. You are not donating plasma. They are giving you money for that plasma, which means that you are selling plasma. Now, in the United States, it is illegal to buy or sell living tissue. Ah, loophole. Plasma is not tissue. Blood is tissue. Plasma is the extracellular fluid. So, you can't sell blood, but you can sell plasma. Now, next rule. If you sell it, if money is exchanged, it cannot go into a patient. So they're not going to take your plasma and go use it directly in a hospital. Why? Because we assume that if there's money involved, you might lie. You'll notice that if you go donate blood in the Red Cross, they ask you a crap load of questions. They're like, have you been out of the country? Have you gotten a tattoo? Did you get a tattoo when you were out of the country? Did you get Ebola when you were out of the country? Do you have AIDS? Do you have Ebola? Do you have AIDS and Ebola? Did you get a tattoo from somebody with AIDS and Ebola? They're super picky about you donating blood because they don't want to give that to a patient and then give them Ebola AIDS. Now, if the Red Cross said, we'll give you 50 bucks for that, you're much more motivated to lie. As is, if you're like, oh, well, yeah, I did get a tattoo from that Ebola AIDS dude, they're like, sorry, we can't take your blood. And you're like, oh, well, I tried. But there's 50 bucks involved. You're like, no, I did not get a tattoo. $50. And then people get hurt. So this plasma, you can't put into a patient. So what do they do with it? Well, they take the proteins out of the plasma and they sell those proteins to a pharmaceutical company who then uses it to make drugs that, yes, may be, important in saving people's lives. Lots of things we can tell from the blood. Hematocrit we talked about. We look at blood glucose. We look at different sizes of red blood cells. We talk about malformations in the blood. Differential white counts give us sources of infection. We can tell the difference between a bacterial infection and a viral infection based on these differential white counts. Complete blood chemistry things. If you've ever looked at that form, like there's a million different things in the blood we can tell based on the ionic composition and the pH. There's lots of diagnostic stuff we can get from the blood. In terms of development, one thing that you do need to know is the difference between fetal hemoglobin and adult hemoglobin. Hemoglobin has a higher affinity for oxygen. Why? Because the fetus isn't breathing. It is completely surrounded by amniotic fluid. So it has to get oxygen at the placenta through diffusion. That fetal hemoglobin has to really, really, really stick to oxygen. It has a higher affinity for oxygen. After birth, we've got hemoglobin A or adult hemoglobin, which still likes oxygen. It has a lower affinity than fetal hemoglobin. As you get older, you're more prone to different types of blood disorders, which are also going to include other disorders, circulatory system, blood vessels, and the immune system. So from here, this would be lecture number 2 on your list. Momentarily, we will talk about some pathophysiology, And you should be looking as well for a blood lab video, which is blood typing, which is a demonstration of the lab that we would do if we were in a classroom. So that's it for this. The next thing that you look for here is your blood typing lab. Look for that next in terms of